Thanks, Kai. So I'm Darcy Diesburg. I'm a graduate student in the Vessel Lab, and today I'm going to discuss the cortico-subcortical beta burst dynamics that underlie movement cancellation in humans. So we've obviously known for a pretty long time now that beta in motoric regions of the brain is tightly related to motor output, both during movement execution and movement control. But recent cross-species research has revealed that on the single trial level, beta is really better represented as a transient and not a sustained signal. So in other words, beta is in fact burst-like. And there's been some work in the past year to begin to connect the dynamics of these single trial beta bursts to what's happening during movement cancellation, specifically using EEG on the scalp. But today I'm going to talk specifically about how we leveraged intracranial recordings in humans to study the relationship between subcortical beta bursts and movement cancellation, and how we tracked the progression of these beta bursts through cortico-subcortical recording sites to also evaluate existing theoretical models of what's happening during movement cancellation. So before I get into the particulars of the project, I of course want to acknowledge important collaborators here. This has been a years-long effort between myself, Jan, and Dr. Jeremy Greenlee, our collaborator in the neurosurgery department. So this work certainly would not have been possible without all of their help. And this project is published as a preprint on the bioRxiv if you're interested in the details, but I'm going to be sharing with you updated results today following a round of reviews. Like I mentioned, we've known for a long time that beta power is tightly related to motor output. During movement execution, we see there's power desynchronization or power reduction, the beta ERD, in regions like the sensory motor cortex, shown here on the left, and in the subthalamic nucleus, shown on the right. But on the other side of the coin, we know that beta power increases are associated with movement cancellation and with motor control more broadly. So Jan has demonstrated that power increases in the STN during action stopping in the beta band, which is shown here on the left, such as during the stop signal task. And we also know that beta power increases occur in the STN during motor conflict, such as during the Simon task, which is shown here on the right. Not only do we see power increases in the STN during the Simon task, we find that beta power in the STN appears to Granger predict beta power in primary motor cortex. And we also know that beta is a marker of differences in unhealthy motor systems. So we see abnormally high beta power in the STNs of individuals, for example, with Parkinson's disease. And when you treat those individuals with synthetic dopamine precursors, of course, we see some degree of alleviation of their symptoms, but also a degree of renormalization of beta power in the STN. So all of this to show that, canonically, we know that beta is tightly related to motor output, but these things that we know about beta have really mostly been contributed by looking at beta as an average over many trials within certain conditions. And as I mentioned, of course, recent cross-species research in rodents, in non-human primates, and in humans has revealed that when we look at motoric beta on the trial-to-trial level, beta really is burst-like and not sustained. So in fact, when we look at single trials and we look at the power on single trials in the beta band, we only see transient increases in beta power that last about 100 to 150 milliseconds. And interestingly, researchers have begun to sort of relate these beta bursts to potential underlying microcircuit mechanisms. So Sherman et al. Demonstrated that these beta bursts likely correspond to simultaneous arrival of proximal and distal synaptic drives to pyramidal neurons. We also have some evidence already to suggest that these beta bursts might index inhibition of processing in neocortex. So Shin et al. Demonstrated that perceptual events that are closely preceded in time by beta bursts are less likely to be detected than those that aren't preceded by beta bursts. And similarly, Simon Little also demonstrated that during movement, if you have some sort of motor movement that's preceded by a beta burst, it's going to be slower than one that isn't closely preceded by a beta burst. So in fact, these canonical beta changes during movement execution and cancellation have to be underpinned at the single trial level by the dynamics of individual beta bursts. And these beta bursts are in fact pretty interesting because we can do things like relate them to underlying mechanisms and to behavior. But the question is, what are the beta burst dynamics during movement cancellation? Well, like again, I already mentioned, there's been some work in the past year to investigate the dynamics of these bursts on the scalp during movement cancellation. And I'm going to highlight some of Jan's work specifically, but I also want to acknowledge that Jana and colleagues also did a study with a stop signal task looking at beta bursts on the scalp. So Jan looked at over 200 scalp EEG data sets that we've collected in our lab with individuals doing the stop signal task. And he found that shortly following the stop signal, topographically, there's an emergence of these beta bursts over frontocentral electrodes very shortly before mean stop signal reaction time. And these beta bursts are more pronounced for successful stops compared to failed stops. When he quantified the beta burst rate of frontocentral beta bursts during stopping, he found that not only were beta bursts increased in the time periods following the stop signal leading up to SSRT, there was a greater beta burst rate at all time bins for successful stops compared to failed stops. And when he quantified the total number of beta bursts between stop signal onset and SSRT, he found that successful stops on average contained a greater number of beta bursts compared to failed stops or match goes. So not only do we see bursts at frontocentral electrodes that do seem to relate to the success of movement cancellation during the time period in which the stopping process is active, pre-SSRT, he also found that these frontocentral beta bursts seem to be related to burst rates in sensory motor cortex. So he found that those trials that contain frontocentral beta bursts, when you saw a burst at frontocentral cortex, it was more likely to be followed within 25 milliseconds by an increase in beta bursts at bilateral sensory motor electrodes C3 and C4. And again, we know that these sensory motor bursts appear to be inhibitory with regards to motor output. So while this isn't a causal demonstration, it does seem that frontocentral beta bursts are followed by an upregulation of beta bursting at sensory motor electrodes. So this study was really interesting and sort of began to paint a picture of what's happening and what role beta bursts on the scalp might play during movement cancellation. But of course, by its nature, this study can't tell us anything about the key subcortical players during movement cancellation and what's happening with beta bursts in those regions. Like Jan has already described, of course, we know that regions like the STN and the thalamus are extremely important for the success of movement cancellation. And it's been proposed that this downstream net inhibition of the thalamus upon thalamic cortical loops is what allows for movement cancellation to occur. But there's currently somewhat of a lack of evidence in humans for this proposed network model. And I'm sure you can understand why this would be the case, because it's somewhat difficult for us to go about acquiring in humans multiple recordings from different regions of the subcortex at once, or really even from the cortex and the subcortex at once. But evaluating this network model is one of the things that we wanted to try to accomplish in this particular study. So our current work had two main aims. First of all, we wanted to determine whether these subcortical beta bursts are related to movement cancellation, and if so, whether they also relate to beta burst rates in sensory motor cortex. And we also wanted to investigate whether the temporal progression of beta bursts throughout these cortico-subcortical recording sites support these longstanding network models of movement cancellation in humans. So we accomplished this by collecting intracranial recordings during awake deep brain stimulation surgeries. We recruited patients who are receiving DBS in either the STN or the thalamus. Those thalamus patients had the ventral intermediate nucleus of the thalamus as the target implant zone specifically, or VIM. So we recruited 9 STN DBS patients, 11 thalamic DBS patients, and we were extremely fortunate to be able to also collect data during a highly unusual surgical case that involved explantation of VIM, DBS, and implantation of STN. So we were actually able to record from both regions at once in that single individual. And in all of these patients, we also recorded from sensory motor cortex, which I've abbreviated SMC here, using subgalial electrode strips. They performed an auditory version of the stop signal task. I won't re-explain the stop signal task here since Berta and Jan have already described it. It's just an auditory version. The go signal is a single sine wave tone in one ear, which cues the side they need to respond to, and the stop signal is a higher pitched beep in both ears that they get on 33% of trials. Before I get into the results, I just want to share a quick note on anatomy, particularly because of our thalamic DBS group. I've mentioned that VIM was the target for implantation here. If you look in the literature, this is called different things depending on which atlas you're referring to. So VIM is also part of VLP. But I also want to mention a second nucleus, which is the ventral oral posterior nucleus, which is a part of VLA. These nuclei are adjacent, but importantly, they receive projections from different regions of the brain. So VIM receives afferents from deep cerebellar nuclei, while VOP receives palatal afferents, such as from the internal segment of the globus pallidus or the basal ganglia. Why am I telling you this? Well, I mentioned VIM is the target zone, but VIM DBS generally tends to be placed on the border between VIM and VOP. So it's likely that these four electrodes we recorded from were spanning both these regions. So I want to make sure you understand that we know that we were recording from motor thalamus more broadly, with not a great ability to distinguish whether it was from VOP or VIM specifically. But I've broken down the results here by the most dorsal contacts on the DBS versus the most ventral. So you'll see me label the most dorsal contacts that I show you, VOP, and the most ventral VIM. So just to get into the results, what we found first of all is that beta bursts decrease across all recording sites during movement execution. So I've plotted beta burst rates on the y-axis, and on the x-axis are time bends in 100 millisecond increments following the go signal. And we can see that beta burst rates quickly decrease in STN, in thalamus, and in sensory motor cortex, while participants are performing movement execution. We also found that beta bursts in our sample as well appeared to be inhibitory with regards to motor output. So we quantified the reaction times on all trials that contained a response, go trials and failed stop trials. And we found that those trials that did not contain a burst between the go signal and the response in sensory motor cortex were faster on average than trials that did contain at least one sensory motor beta burst. Again, evidence that these bursts do seem to have an inhibitory effect on motor output in sensory motor cortex. Our main question, of course, was how do beta bursts in the subcortex relate to movement cancellation? So we quantified the total number of beta bursts between stop signal onset and stop signal reaction time. And we found that successful stops, shown here in light blue, on average contain a greater number of bursts during stopping compared to matched go trials, and in the STN and the VIM compared to failed stop trials as well. We also quantified beta burst rate, again in time bends following the stop signal. But prior to SSRT, we didn't find pairwise differences for particular time bends the same way we did with total beta burst count, which to us just suggests a pretty tight relationship between participant-wise stop signal reaction time and these stop-related beta bursts. We did find late increases in beta bursts post-SSRT, which I'm going to come back to later to try and explain. But we found that beta bursts in the subcortex, again, do seem to relate to movement cancellation pre-SSRT during the time period in which the stopping process would be active. The next question is whether they seem to also relate to sensory motor cortex beta bursts. So we time-locked beta bursts in sensory motor cortex to beta bursts happening in the subcortex. And we found that sensory motor cortex bursts increased in rate within 50 milliseconds following STN bursts. Interestingly, this was not the case with bursts in the thalamus. And this also wasn't the case when we looked at this the other way around, looking at subcortical bursts locked to sensory motor cortex bursts. The next aim of our study, of course, was to try and investigate existing network theoretical models by looking at the progression of beta bursts throughout subcortical regions we've recorded from. If we see results that align with these existing models, we might expect to see STN bursts emerge first, followed by bursts in the thalamus, again suggesting the STN is recruited first and might then net inhibit the thalamus. And this actually is in fact what we found. So we calculated the timing of first bursts in subcortical regions, particularly looking at this with respect to individual participant stop signal reaction time. On the sample level first, understanding of course that STN recordings and thalamic recordings came from different individuals, but what we found is that during successful stops, bursts tended to occur earlier in the STN compared to bursts in the thalamus. Now, again, these were different subjects, of course, so the main test of this will be to look at this timing in our single subject that had recordings from both the STN and the thalamus. And we found the same timing difference in that single individual. So we found that during successful stops, bursts in the STN do emerge first, followed by bursts in the thalamus, and specifically followed by bursts in the thalamus in the dorsal most contacts. So those contacts that I have here labeled VOP. And this is exciting to us, not only because it's highly unusual to be able to record from multiple subcortical regions at once in humans, but also because this seems to align with this network model that the STN might net inhibit the thalamus and potentially thalamicortical loops. So to sum up what I've shown you, we found that pre-SSRT beta bursts in subcortex do seem to relate to movement cancellation, that STN bursts are followed by an upregulation of bursts in sensory motor cortex during cancellation, and that STN beta bursts do seem to precede thalamic bursts during cancellation in both the sample and in our single subject with recordings from both regions. Now, of course, the one result I haven't explained to you so far is this one I said I'd come back to, which is the fact that we did find post-SSRT beta bursts for successful stops in all of our subcortical regions. And of course, we didn't have a hypothesis about this specifically, so we just attempt to offer an explanation, understanding, of course, that future research is needed to corroborate this. As Jan described, a pause then cancel model of inhibition does predict that both the hyperdirect and indirect pathways required for successful inhibition. So we think it's possible that these later peaks in beta bursting might reflect just the slower engagement of the indirect pathway. And if this is true, then this might be that transient beta is a signature of inhibitory signaling in both pathways. But again, we need more research to corroborate this account. I want to finish with just some explanations of why this is interesting for those of us who are interested in cognitive control more broadly. So I've talked about this fronto-basal ganglia network today, mostly focusing on its role in movement control, but we know that the same fronto-basal ganglia network also interrupts cognitive processes following surprise, such as resulting from novel or infrequent events. So for example, we know that surprising events seem to interrupt verbal working memory and ongoing perceptual processing, ostensibly by way of the same neural circuit. So Jan demonstrated that presenting an unexpected bird sound in a verbal working memory maintenance period leads to hits on working memory accuracy. And Cholso in our lab demonstrated that ongoing perceptual processing, as indexed by steady state visual evoked potentials, is similarly interrupted by the presentation of unexpected sounds. So the question then is, do beta burst dynamics during cognitive control have similar characteristics as they do during motor control? And if they do, this leads to potentially interesting avenues of research where beta bursts might serve as a single trial level signature of behavior, of potential mechanisms during instances in which cognitive control is needed, such as during surprise, following error, and following unexpected action outcomes as well. So I just want to thank a few people who, again, were a big help during this project, especially Jan, lab members Nathan and Tobin, who helped with some data collection on this project, our collaborators in the Department of Neurosurgery, Dr. Jeremy Greenlee and Haiming Chen, the patients for being willing to participate in research, and our funding sources. And thank you for listening.